Hello, I'm Dr. Jeffrey Hayes, Education Specialist of the American Society for Reproductive Medicine. Welcome to this presentation of the ASRM Grand Rounds webinar series. These webinars are designed to address topics in the ABOG Learning Guide in Reproductive Endocrinology and Infertility. Today's presentation is by Dr. Kyle Orwig. Dr. Orwig is Professor of OBGYN and Reproductive Sciences at the Magee-Womens Research Institute, University of Pittsburgh School of Medicine. The title of his talk is Spermatogenesis and Male Fertility. I will now review the details of today's presentation. After the webinar is done, please do not forget to return to ASRM eLearn to take the post-test and complete the survey for your continuing education credits. You must complete the post-test question successfully and complete the survey to receive credit and be able to print off your certificate. If you wish to ask a question to the speaker about today's presentation, when you return to ASRM eLearn, click on the page link labeled Questions and an email address will be provided. We are very excited for our talk today, so I will now turn things over to our speaker.
Hello everyone, welcome to this webinar on spermatogenesis and male infertility. As I was introduced, I'm a professor of OBGYN and Reproductive Sciences at the University of Pittsburgh. I'm also the Director of Research in the Reproductive Endocrinology and Infertility Division and the Director of the Fertility Preservation Program. So my first essential slides. First, I have nothing to disclose. And our learning objectives today are that the participants should be able to understand the basis of male infertility, which includes being able to explain spermatogenic lineage development. Participants should be able to review the anatomy of the testis and also explain the role of gonadotropins and testicular somatic cells in the regulation of spermatogenesis. And finally, participants should be able to recognize normal semen parameters in the fertile male. With respect to male infertility, the participants should be able to state the prevalence of infertility as well as some of the diagnoses that are made in the male infertility clinic. We will review some of the causes of infertility, and finally, the participants should be able to describe some of the treatments for azoospermia, and that includes both standard of care as well as experimental. So spermatogenesis arises from a population of stem cells called spermatogonial stem cells, or SSCs, and these stem cells are defined like every other stem cell type that I know about, and that is by two activities. One is that they self-renew to maintain the stem cell pool, and the other is that they differentiate to give rise to one or more lineages. In the case of spermatogenesis in the testis, there's exactly one lineage, and the terminally differentiated product is the spermatozoa that carries the haploid genome and is able to swim through the female reproductive tract. So once the diploid spermatogonial stem cell commits to differentiate, it produces a progenitor spermatogonia that undergoes some species-specific number of transit-amplifying mitotic divisions before producing the primary spermatocyte that then undergoes two meiotic divisions to reduce to the haploid genome, and then the morphogenesis or spermiogenesis that produces the terminally differentiated sperm. There are differences between species, so when the spermatogonial stem cell in a mouse commits to differentiate, the resulting spermatogonia undergoes a total of 10 transit-amplifying divisions, followed by 2 meiotic divisions before spermiogenesis occurs. So with a total of 12 transit-amplifying divisions, a single stem cell that commits to differentiate can theoretically produce up to 4,096 sperm. The situation is a little bit different in men because in men there are only 1 or 2 transit-amplifying divisions that occur at this stage here, and so that means that a single stem cell that commits to differentiate will theoretically produce only 16 or 32 sperm. But the men in the audience shouldn't despair because you are still producing 4.4 million sperm per gram of testicular tissue per day. and the average testis size of a man is about 15 grams. So with 2 testes, a man produces about 132 million sperm per day. And assuming a heart rate of 60 beats per minute, that means that a man is producing about 1,500 sperm every time his heart beats. Well, all of this process of spermatogenesis occurs here inside the seminiferous tubules of the testis, where each seminiferous tubule both begins and ends in this structure here called the reedy testis. So sperm are produced in the tubules, they flow out into the reedy testis, and then through the efferent ducts into the head of the epididymis, where they become more mature and acquire the ability to swim. If we take a cross-section through one of these seminiferous tubules, we would find that the stem cells and their immediate progeny are all located down here on the basement membrane of the seminiferous tubules. And then, as these spermatogonia differentiate to produce spermatocytes and enter meiosis, they move away from the basement membrane and differentiate until eventually mature sperm are released into the lumen of the seminiferous tubule. So next we're going to zoom in on a segment of the seminiferous epithelium to look at it in more detail so that we can understand this somatic environment where spermatogenesis occurs and also the endocrine regulation of the testis. So here you're looking at a cutout of a seminiferous tubules where the germ cells are shown in blue, including the most undifferentiated spermatogonia located down here on the basement membrane. Then, as I said, those cells move off the basement membrane and enter meiosis to produce spermatocytes, spermatids, and eventually terminally differentiated sperm that are released up here into the lumen. Well, spermatogenesis can only occur because there are several critical somatic cell types that are located in the testis that regulate sperm production. And the one somatic cell type that is located inside the seminiferous tubule is the Sertoli cell. That's shown here in the salmon color. Sertoli cells are amazing cells that stretch all the way from the basement membrane of the seminiferous tubule up to the lumen, and they regulate every step of spermatogenesis from the most undifferentiated spermatogonia to the most terminally differentiated spermatozoa. And on the right-hand side here, you can see artist rendition of a scanning electron micrograph of a Sertoli cell. It's a very complex cell type, and I think you can even imagine the germ cells sitting in these pockets of the Sertoli cells that nurture every stage of germ lineage development. The other important somatic cell type in the testis that we'll talk about today are the Leydig cells. These are the source of testosterone, and these are located outside of the seminiferous tubule. Well, just like the female, the testis in males is regulated from the brain by the production of gonadotropin-releasing hormone from the hypothalamus that stimulates the release of luteinizing hormone and follicle-stimulating hormone from the pituitary. At the level of the testis, follicle-stimulating hormone acts on Sertoli cells, and it regulates important functions, including the proliferation of Sertoli cells that occurs during puberty, and also the cyclic regulation of spermatogenesis that occurs throughout the postpubertal life of men. Luteinizing hormone, or LH, that's produced from the pituitary, acts on the Leydig cells, and these are the source of testosterone that is absolutely essential for spermatogenesis and regulates every step of spermatogenic lineage development. And testosterone also acts back on the brain to regulate the production of both FSH and LH. If all of this process of spermatogenesis works correctly, then the World Health Organization tells us that a normal semen sample should include 1.5 mL of semen per ejaculate, and in that ejaculate, the sperm concentration should be 15 million sperm per milliliter. And if you multiply the volume times the concentration of sperm, the overall number of sperm in an ejaculate should be about 40 million sperm. But we know that spermatogenesis doesn't occur correctly all the time. 10 to 15% of couples in the United States are infertile, and a full 1% of men in the general population are azoospermic, which means they are not producing any sperm in their ejaculate. And in the United States, that translates to 645,000 men between the ages of 20 and 50, which you might consider to be their prime reproductive years. Well, azoospermia can occur in two varieties. In 15% of cases, azoospermic is due to an obstructive condition. And what that means is that even though these seminiferous tubules are producing sperm, there's some blockage in the excurrent duct system that prevents the release of sperm into the ejaculate. This condition is relatively easily treated because the urologist can simply take a biopsy from the testis or aspirate sperm from the epididymis, and then those sperm could be used in the IVF clinic using intracytoplasmic sperm injection to achieve fertilization and a pregnancy. But in the majority of cases, 85%, azoospermia is non-obstructive. And what that means is that the excurrent duct system is functioning normally, and that probably there's a defect in spermatogenesis here inside the seminiferous tubules. But even in those cases with the most severe diagnosis of non-obstructive azoospermia, it is sometimes possible to retrieve sperm directly from the testes. There are a variety of approaches for doing this, and one of those approaches is called microsurgical testicular sperm extraction, or microtesty, and this has an average success rate of about 50%, which is pretty impressive. And to give you an idea how this works, I've colored some of the seminiferous tubules here in the testis to indicate that for some reason there are small patches of tubules that still contain spermatogenesis. And so if the urologist can access these patches and extract them from the testis, then it might be possible to retrieve sperm that can be used for reproduction. So this slide shows the microtesty procedure, where an incision is made in the tunica that surrounds the seminiferous tubules to reveal all the seminiferous tubules of the testis. And if we zoom in microscopically, I think you can appreciate that some of the tubules are very skinny. That's because they lack spermatogenesis. But occasionally you can find an island of fat seminiferous tubules, and the reason that these seminiferous tubules are so plump is because they're filled up with spermatogenesis. So the urologist can remove these segments of seminiferous tubules and tease the tubules apart, and oftentimes, in 50% of cases, it's possible to retrieve sperm from those segments. So I just told you about the technique of microtesty, which is indicated here on the left-hand side. Like I said, the success rate of microtesty for sperm retrieval is about 50%. But there are other methods to retrieve sperm from the testis. One is a wedge biopsy. So in this case, I've taken the liberty of marking two tubules with a black color, which indicates that this is where the plump seminiferous tubules are located. And as you can see, depending on where one takes the biopsy, the chances of identifying the plump tubules in a single biopsy like this might be relatively lower than it would be in the microtesty technique. However, this procedure is much quicker than the microtesty technique, which can take 3 to 4 hours to find the tubules with spermatogenesis. Another option is to use a needle biopsy to extract tubules from the testis, and you can imagine the chances of hitting the plump tubules this way might be even less than a wedge biopsy. And the final method I'll tell you about is one that was pioneered from Paul Turek's lab, and this is a technique called fine needle mapping of the testis. And here what you can see is that several biopsies are taken from the testis, and each one of those biopsies is analyzed by the andrologist. Then when the andrologist reports back to the urologist that sperm were identified in this biopsy and this biopsy, then the urologist can go in and make a wedge biopsy here and have a higher chance of retrieving sperm. So what's important to know is that being infertile, that is, for example, being diagnosed with non-obstructive azoospermia, does not mean unable to conceive, and that's because we know that sperm does not have to traverse the male or the female reproductive tract to fertilize an egg, and that we have several technologies in the assisted reproductive technology or IVF laboratory that we can use a relatively lower number of sperm to achieve fertilization in a pregnancy. And those options include intrauterine insemination. This ideally requires between 10 and 20 million sperm, but is possible with even fewer. And in this case, those sperm are deposited directly into the female reproductive tract to fertilize the egg in the usual way. If the number of sperm available are even lower, then we have the option of in vitro fertilization, where about 50 to 100,000 viable sperm are required per oocyte to achieve fertilization. And finally, if the number of sperm are particularly low, we have the option of intracytoplasmic sperm injection, where of course it only takes one sperm to fertilize an oocyte, and even a sperm that is of relatively lower quality or not able to swim in the normal progressive fashion still can be used because we use a needle to insert the sperm into the egg. So I'm just going to show a short movie of intracytoplasmic sperm injection, which I'm sure many people have already seen, but illustrates the point that in order to achieve fertilization in a pregnancy, we need exactly one sperm and one egg. So here you can see a single sperm in the injection pipette, and I'll let this movie run. So first, some of the egg cytoplasm is drawn back up into the needle, and then the sperm and the egg cytoplasm are ejected and left here. So this slide shows an early study from Peter Schlegel, who is one of the pioneers of microtesting in the United States. This is a study in which they recruited 81 patients with that most severe diagnosis of non-obstructive azoospermia, and amazingly, they were able to recover sperm in 58% of cases. The fertilization rate using that sperm was 61%, and the overall pregnancy rate was 32% in that particular study. So now I'm going to sidetrack just for a moment to remind you that Louise Brown was the very first baby produced by in vitro fertilization. She was born on July 25th of 1978. And on the left here, she's shown in a picture with her dad, John, and her mom, Leslie. And on the right-hand side, she's holding the incubator where her dad's sperm met her mom's egg and gave rise to Louise. So this now is a very well-established technology that has produced about 6 million babies worldwide. And I think what's exciting is that that technology led to the 2010 Nobel Prize in Physiology and Medicine. And this was for collaborative work done by Dr. Patrick Steptoe, who was the physician, and Bob Edwards, who was the researcher. Bob Edwards was eventually the recipient of the Nobel Prize. Well, what's important to know is that Louise was possible because her dad was able to contribute a sperm, and her mom was able to contribute an egg, and that her mom had a uterus that was receptive to embryo implantation and embryo development. However, as I indicated before, a full 1% of men in the global population and also in the US population are azoospermic, and we're only able to recover sperm in about 50% of those cases using the technique of microsurgical testicular sperm extraction. So that leaves another 50% of patients who cannot be treated for their infertility if they have a diagnosis of azoospermia. Well, there are a variety of conditions that can cause azoospermia. Those are reviewed on this slide here. The most common is varicocele, and you'll see a variety of other conditions that can lead to infertility on this slide. But for the next couple of slides, I'm going to focus on infertility that can occur due to gonadotoxicity of a medical treatment, such as radiation or chemotherapy for cancer or other conditions. And the reason that I'm going to focus on that is because we know that chemotherapy and radiation can cause permanent infertility. and because cancer survivors report back to us through epidemiological surveys that their fertility status has an important impact on their quality of life. And so for that reason, the American Society for Clinical Oncology, the American Society for Reproductive Medicine, and many other of our learned societies recommend that all men and women be educated about the effects of their medical treatments on their future fertility, and also about the options for preserving their fertility before they start treatment. And for adult patients, those options include cryopreserving eggs, or cryopreserving sperm, or cryopreserving embryos, which can be used in the future to achieve pregnancy using assisted reproductive technologies. But these options are not available to pre-adolescent boys and girls because they're not yet producing mature eggs or sperm. And this is an important human health problem because the 5-year survival rate for kids with cancer has now surpassed 85%, and that's due to the rapidly improving therapies for childhood cancers. And so what that means is that we as a medical community are producing a lot of survivors who still have their entire reproductive life in front of them. Well, in the case of the prepubertal boys, even though they're not making sperm, we do know that they contain those critical spermatogonial stem cells in their testes that are poised to begin producing sperm at puberty. And now there are several academic centers, both in the US and abroad, that are actively preserving testicular tissues for boys, also ovarian tissues for girls, because we anticipate that we'll have new reproductive technologies available for those patients in the future. So I'm going to finish up this webinar today with a few thoughts about what might represent the future of reproductive medicine. So in the case of the boys that froze testicular tissue in advance of a chemotherapy or radiation treatment, the options that are available to them, first and foremost, is to cryopreserve a semen sample. This, of course, is only available to a teenage boys or adult men. But what's important to know that if it is possible to save a semen sample, that is the appropriate approach and the standard of care approach, because we know that sperm make babies. And based on what I've shown you already in this presentation, I hope you understand that the semen sample does not have to be an outstanding semen sample, because using intracytoplasmic sperm injection, it only takes a single sperm to fertilize an egg. However, for those patients where collection of a semen sample is not possible, for example, the prepubertal boys, we have the option to take a testicular biopsy from the testis. And like I said, there are several centers around the world who are actively freezing testicular tissues for boys. And the reason for that is because we think that in the future we may have either cell-based therapies or tissue-based therapies that can either regenerate normal spermatogenesis back in the testis or produce sperm that can be used to fertilize an egg with ICSI. And looking perhaps even further in the future, one day it might be possible to take a skin biopsy or a blood sample and convert those samples into what we call induced pluripotent stem cells, or iPSCs, that then can be converted either into transplantable spermatogonial stem cells or into haploid sperm that can be used to fertilize an egg by ICSI. Well, all of the options on the bottom of this slide may look a little bit like science fiction, but what might be surprising to you is that actually every method on this entire slide has produced spermatogenesis and live-born babies, at least in mice. And some of these techniques are actually relatively mature, including spermatogonial stem cell transplantation and testicular tissue grafting. These methods have been replicated in several mammalian species, including non-human primates. So these are two technologies that are nearly ready to translate to the clinic now. So I've given you an example where the cause of infertility was known, that is, to a medical treatment, but it turns out that the majority of infertile patients we're not able to determine what the cause of azoospermia was. So this shows you a pie chart of patients with uniform maturation arrest. Patients that have this condition have germ cells in their testes, but they're not able to mature all the way to sperm. And if this is a uniform condition, then the chances of recovering sperm using microtestis are relatively low. And the causes of uniform maturation arrest or a more severe diagnosis of Sertoli cell-only syndrome include deletions of the Y chromosome, which explain nearly a quarter of azoospermia. We also have other karyotypic abnormalities, and other conditions include, for example, medical treatments that I already told you about. But importantly, over 50% of cases of azoospermia eventually get a diagnosis of idiopathic, which simply means we don't know what the cause of infertility is. And the thought in the field is that about half of those cases might be due to a genetic defect. And this is based on observations in animal models. In the future, it is possible that we may be able to discover the genes that cause infertility and to develop the therapies that could restore fertility in those patients with idiopathic infertility. And that future is coming closer to us now due to a few things that have happened in the last several years. One is that the cost of sequencing the human genome, we've nearly achieved the goal of doing that for in the neighborhood of $1,000, which means that it might be accessible in the medical clinic. And if we're able to sequence the genomes of patients, it may be possible for us to discover the genes that cause infertility. And if we do that, it may be reasonable to develop screening tools so that we can diagnose the cause of infertility in patients that currently get an idiopathic diagnosis. And finally, if we're able to discover the genes that cause infertility, then in the future we may be able to use advanced gene editing technologies such as CRISPR-Cas9 to precisely modify the genome and repair the genetic defect that led to the infertility. This is something that is some distance in the future. Important safety and feasibility studies still need to be done before this can be considered in the human clinic, but hopefully some of you will be deploying these in the clinic in your future careers. And that concludes my webinar. Thanks for your attention, and I'll be happy to answer any questions as they arise.